Hello and welcome to Nara.com, and today we're gonna discuss the real causes of cancer, and not based on beliefs but based on scientific facts that we're gonna present to you. We're gonna discuss the mainstream theories on how you get cancer and then present some very well researched alternative theories that are not so well known, and much more. And in the end, of course, we are going to talk also about some holistic healing approaches. So without further ado, let's learn more about the real causes of cancer, whether it is a viral infection that causes cancer, genetic mutations, or indeed a microparasitic infection. And why we need to discuss this subject shows you this table, because as you can see, from 1990 to 2015, the cases of cancer per year, per day, the deaths per year, for example, have increased from approximately 510,000 deaths per year in 1990 to almost 590,000 in 2015, and the numbers are still on the rise. And so the question arises why we have this epidemic in spite of the tremendous progress in medicine, the billions that have been invested into research and development work by the pharma industry, as well as the cancer associations around the world, etc. And still the numbers are on the rise, as you can see, and it seems that we have no real cure for cancer still. So why is that so? Is it possibly because we are following a concept that is not completely correct? And this is what we're going to try to answer today. So let's answer. What's wrong? Why is the number of cancer deaths permanently increasing? What are the real causes of cancer, and what must be changed in the standard healthcare procedures to reduce the number of people who are suffering from cancer dramatically, as well as, of course, reduce the number of deaths? And I will give you here 4 answers. The first one is that obviously the number of new cancer cases as well as the number of cancer deaths come about because of a wrong non-efficient therapy. That is an option. The second option might be that the 3 major theories how cancer develops in your body, which are: first theory is genetic mutation. We're going to discuss all of those a little bit more in depth in the following slides. Then the metabolic mitochondrial respiration disorder might be a cause according to mainstream theories. It also, another theory suggests that it comes about due to a viral infection, and also the predominant dogma is that of the genetic mutation, as you will see. The third option is that there are numerous alternative medical treatments that can clearly improve the patient's life quality and they can expand their lifespan. However, we have not yet discovered a real panacea, so a real cure for cancer. And cancer standard healthcare procedure follows always the same schematic. You remove through surgery the cancerous tissue, and you avoid a recurrence of tumors using chemo or radiation therapy. And the result is that over 90% of patients that are treated this way do not survive cancer by more than 1 to 5 years without additional complications. And on top of that, during the treatments, they face a miserable quality of life. So let's see what is wrong with the existing mainstream cancer theories, and if we can find a well-researched, better alternative theory that explains why we haven't had success with our current treatments. And then, of course, last but not least, let's find out if there are some holistic treatments with very little side effects or none that really work and that can really help you. And I would like to start with the most widespread, most popular theory about how cancer develops in your body, and this is called the genetic theory. So the mainstream media and the medical establishment promote this concept of cancer being a genetic disease, and this is how they explain it. The cells in the human body work together and they divide and reproduce in a strictly controlled and coordinated way. And the DNA replication is an essential part of the cell's division. And before they divide, the cells make an identical copy, a blueprint of their genome, which is stored in the RNA. Mutations then can occur during this DNA replication, which in turn changes the genome of the new cells. And most of these copying mistakes, so to speak, are quickly repaired by the healthy cells if the organism is working properly. But sometimes mistakes do change those DNA blueprints. And if these mutations occur in the genes which control the growth, which are also called proto-oncogenes, and the tumor suppressor genes as well, the result is an uncontrolled cell growth, and this leads to cancer, according to this theory. The proto-oncogenes encourage the cell to multiply, and if these genes become mutated, they instruct the cells to multiply all the time, and these are then called oncogenes. The tumor suppressor genes, on the other side, they stop the cell from multiplying, and if these genes become mutated and stop working properly, the cells may carry on the cancer proliferation process. Mutations then accumulate over many years before a cell becomes cancerous, and this is why cancer is more common in elderly people. Initially, the tumor is confined to the tissue in which it is located, but as the tumor grows bigger, it requires more nutrients, so it starts to develop its own blood vessel network, and this process is called angiogenesis. The cancer cells then develop the ability to migrate also via the bloodstream or the lymphatic vessels, and they create then what is called a metastasis. Some individuals inherit also genetic predisposition for certain cancers according to this theory. And this graph shows you really well how this whole theory works in your body, exactly as we've discussed it, but here in a visual display. Sounds really logic, this whole genetic mutation theory, doesn't it? So what is the problem with it then? Well, the problem is that experiments that were performed already in 1969, for example, by McKinnell and others, and were published in 1969 in Science magazine, showed that this cannot be 100% true. It cannot be the real cause of cancer, because what they did in these experiments was they took already tumorous renal tumor cell from a frog, and they cloned it, and the transplant tadpole that resulted from it was not tumorous. It had a well-formed head, a body, and tail, as you can see here from the pictures. On the left, you see the tumorous, the renal tumor from the original frog. They took it, cloned it, and what was expected according to this genetic mutation theory was that the tadpole should also be tumorous, or at least mutated in a cancerous way. But the result was a perfectly well-formed tadpole without a tumor. So, questions. Why is that so? If this whole genetic mutation theory is correct, then the tadpole should also have been cancerous, or at least misformed. Another theory is that cancer is a metabolic disease. In 1956, Otto Warburg discovered the so-called Warburg effect, and according to this, the cancer arises from damages at the level of cellular respiration that happens in the mitochondrial cells. The normal energy produced by a healthy cell per one molecule of glucose is 36 ATP, equal to 56 kilojoules, and this is generated during the glycolysis of food and the oxygenation of the pyruvate during a healthy mitochondrial respiration. And when cancer rises, according to this theory, this energy is diminishing because the digestion switches to a fermentation mode, which is a very primitive way of creating energy that is characteristic for single-cell organisms, so primitive, less differentiated organisms, and cancer cells continue to ferment lactate in the presence of oxygen. And so, according to this theory, the fermentation collapses the ATP energy in the cells. In cancerous cells, the glucose uptake and the glycolysis proceed about 10 times faster than in non-cancerous tissue, and tumor cells commonly experience hypoxia, which is a lack of oxygen, because they initially lack an extensive capillary network to supply the tumor with oxygen that is brought by the bloodstream. And the high glycolytic rate may also result in part from smaller number of mitochondria in the tumor cells that generate less ATP. So the cancer cells literally kidnap the ATP from the healthy cells. In some cases, the tumor cells overproduce several glycolytic enzymes, committing the cells to continue the glycolysis. And HIF1 is a protein that stimulates the activity of 8 glycolytic enzymes and give the tumors the capacity to survive anaerobic conditions, or conditions with a lack of oxygen. And therefore, most chemo drugs and cytostatic remedies of all kind must generate in situ ROS, which is reactive oxygen species. Another theory that was put forward in the 1970s is the so-called viral infection theory, according to which the cancer is a result of a virus or fungal infection. Viruses, hypobiotic protein fragments penetrate, according to this theory, the healthy cells and interfere thus with the host's cellular metabolism, and through the host cell mitosis the viruses can multiply and the tumors start growing as a result. Problem is that today we have electron microscopes with up to 500 times magnifying power, and in spite of these advanced tools, viruses have never really been characterized, measured or separated in any experiment. Most of the pictures that you see many times of, for example, the Ebola virus or HIV virus or hepatitis B or C virus are simple pictograms, graphic representations, and not real photographs. And that's why we would suggest that this whole theory is incorrect. There is a lot of money, of course, behind it, because you can vaccinate against viruses and you can give antibiotics, and this, of course, can create a lot of income. That's why probably this theory is still around. Viruses are actually only tiny protein fragments that result from the asexual microparasites or the bacterial proliferation. They are literally a piece of dead flesh, protozoa genetic material protein. But the cancer virus dogma is very easy to combat with existing experimental methods, and it has never been scientifically demonstrated that it's correct. So we can strike the genetic mutation theory off the list. As we've seen, we can also strike off the list the viral infection or fungal infection theory. So what other theories have been put forward over the years which might be correct or not? Well, another one is the so-called microparasitic infection theory. Between 1965 and 1990, Dr. Alfons Weber and many other renowned biologists, doctors, and renowned research and development institutes such as Pettenkofer Institute, Koch Institute, Pasteur Institute, they developed and experimentally demonstrated that cancer is a microparasitic, a protozoan infection that starts in the red blood cells. Dr. Weber's work is based on thousands of biological in vitro and in vivo experiments with tissues and blood samples collected from cancer patients during their life and even after their death. And in all of these tests, all blood and tissue tests confirmed that the microparasites were present in the blood plasma as well as the cancerous tissue of those patients. The cancer theory that it based on this microparasitic infection is exceptionally well and accurately documented. Dr. Alfons Weber did not only theorize about that, but he filmed live hundreds of his tests and the blood samples. He did thousands of measurements in different cancer stages, and we have already a very interesting live footage video up on our a YouTube channel that is called Video Proof Cancer is a Microparasitic Infection, where you can see all of this live footage, or at least a sample of it for yourself, and what really happens in the cells of the cancer patients. Microparasites as the single real cause of cancer. This theory has been widely supported by many studies and experiments that have been performed by renowned biologists and doctors such as the Robert Koch Institute, the Pasteur Institute. Tamara Lebedeva did experiments on this as well and confirmed the theory of Dr. Weber. Max from Pettenkofer Institute performed similar tests with the same results. Today we still have the University of Munich, Germany, the medical faculty, who are still performing experiments in this direction, and they still get the same results. Dr. Alan Cantwell, Professor Dr. Peter Duisberg, Sir Henry Britlin, Wilhelm Reich, many doctors, they performed these experiments because Dr. Weber, he published all of his methodology. You can go online and find it, and you can exactly replicate this test for yourself. It's not difficult either. You take a blood sample, you don't need a special microscope even, and dark field microscopy, you will clearly see what is going on in the red blood cells. So the conclusions after all of these studies are that the protozoa are more pathogenic than any bacteria and fungi together, and they are the real cause of cancer and other chronic diseases. Okay, so this theory with microparasitic infections looks really promising already. Let's see if we can also explain some of the unanswered questions that we have put forward at the beginning of this presentation. Like, for example, can we explain with this theory the exponential spread of cancer from 1950 onwards? And one of the microparasitical infectious carriers is the meat and bone meal that is used as cattle fodder and thus present in any cattle or poultry meat. And these so-called merozoites, the genetic materials of the microparasites, they can survive heat, cold, as well as chemicals, so they are extremely resilient. Microparasites are the primary and oldest life form on this planet, and their adaptation capabilities are very advanced, much better than ours actually. And the same microparasitically infested genetic material is abundantly present in poultry excrements, in manure, which is then also spread as a fertilizer, so it also gets into our vegetables, fruits, etc. Through the international traffic, the globalized world that we live in, with all the traveling and the goods that travel from continent to continent, and our globalized food chain supply, as well as the mechanized and very intensive fertilizing with animal excrements, and or the feeding industry that uses, as we've discussed, bone and meat meal, which is all already infected with this genetic material of this microparasites called protozoa. Through excessive vaccination, use of antibiotics, blood transfusions, as well as direct insect and animal bites, these microparasites can spread epidemically around the globe, and either the insect directly infects the blood capillary, or the infection happens through the lymphatic tract and the parasites penetrate into the bloodstream later. These microparasites that are plasmodia-like and are called protozoa, as mentioned, are very difficult to identify in blood lab work because they hide inside of the red blood cells, the so-called erythrocytes. But as you can see in our video about the cancer proof provided by Dr. Weber, with certain not very expensive techniques, you can make it visible. The environmental toxicity, the electrosmog burdens, the GMO and the poor food quality, as well as stress and psycho-emotional overload that weakens our body, destroys the blood capillaries and penetration. Therefore, we become more susceptible to the penetration and spreading of the very resilient microparasites. So the question of becoming infected is not if, but when, because when our organism is sufficiently weakened, these microparasites that can be dealt with very well by a healthy immune system, as you can also see in the live footage, cannot deal with the number of parasites anymore, and then we have a real probability for developing chronic diseases as well as acute ones, and worst case scenario, even cancer. Protozoa are defined as a group of unicellular eukaryotic organisms, and they are single-celled organisms with animal-like behaviors such as motility and predation, nutrition and excretion, as well as sexual proliferation. And the members of the protozoa are the ciliates, the amoebae, and the flagellates. What is also very important to understand is that the protozoa are not viruses, bacteria, or fungi species. They are one single cell living organism that are the first form of life that have developed on this planet, and one can say that microparasites are the beginning of life on Earth, therefore. And in turn, these minuscule animals are so resilient and well adapted, it's incredible. They can independently move around, they may have a suction cup organ, they have their own metabolism, they use the same gases as animals and humans, which are oxygen and nitrogen, of course, to survive. They feed themselves with glucose and proteins and use water and other fluids. They absorb also light and sound. They have a cellular membrane. They are spraying many toxic substances into their host cells and around the connective tissue. With isolated protozoa, you can also induce cancer in animals and humans, as experiments have found out, opposite to tumor cell cloning, which does not produce a cancerous tumor, as you have seen in the frog experiments earlier on. The protozoa can be grown in fertilized chicken eggs. They proliferate through mitosis and asexual amitosis at a very high rate. They use the human body cell's energy, and thus the membrane potential decreases from 70 to 100 millivolts to 15 and even lower than that, which is of course not good because they deprive you of the energy that you need. The protozoans range in diameter from a few thousandths of a millimeter to several millimeters. So those are real interesting creatures. And unfortunately, if our body does not have the capabilities, because It's been bombarded by all kind of toxins, etc. So it's out of balance if when it does not have the capability anymore to deal with those creatures, we get a lot of problems as a result. What are some of the symptoms of this microparasitical invasion? Well, blood quality will change, so the viscosity increases, the erythrocytes clump and lose their biconcave shape, and the cell's superficial tension decreases due to changes in electrical charge. The oxygen transport is therefore severely hampered, and the cells eventually die. The blood circulation is also significantly impaired. The infected erythrocytes lose hemoglobin, so they go into anemic state, as well as the glucose, plasma, protein serum, and the cholesterol. All these substances are now missing in the host's metabolism, and this is why cancer patients have also very often diabetes 2, anemia, or even leukemia, high cholesterol values, as well as globulin values. And they are also cyanotic, undernourished, tired, and fatigued, and have a toxic metabolism. Hyperplasia is another symptom. It is boosted to enhance the stem cells division and proliferation, followed soon by hypoplasia, and leukemia is one of the consequences of blood hypoplasia. Erythrocytes and lymphocytes counts are also diminishing. Dead cells and cellular debris are swimming around in the bloodstream. The capillary permeability is reduced and the general capillary circulation is impaired. Increasingly, the gases cannot be purged, therefore oxygen cannot permeate the capillaries, and blood is under-oxygenated as a result. Also, the cellular debris cannot be excreted, so the patients become then toxic. Infected blood plasma is also leaking out into the connective tissue, so extravascularly, and into the organs therefore, and the protozoa are now infecting the lymphatic vessels and lymph nodes, the endocrine glands, the cartilages, as well as all of the organs, and this is called stage 2 and 3 of the extravascular compensated. Blood vessels are either too dilated or too constricted or obstructed also. Some blood vessels are completely blocked. Aneurysms may appear to support blood circulation. Other side effects of these plasmodial infections are microinfarctions, necrosis, obturation, chronic heart insufficiency, heart arrhythmia, valve insufficiency, and may go also as far as a heart attack or stroke. The microparasitical invasion of the lymphatic system leads also to a swollen, painful lymph node, which is also called lymphadenitis. So how do these microparasites create cancer? Well, how do the stem cells proliferate and create tumors when they become infected with the microparasites would be the more precise way of putting these questions, because the mutated cells are the result of the microparasitical invasion of the stem cells. What do the stem cells do when they become invaded by those microparasites? Well, to get rid of the overwhelmingly invading protozoa, the stem cells start to over-proliferate in order to dilute the number of invaders per cell, and you can see this very well in the video by Dr. Weber. And the parasites invaded stem cells lose then the correct genetic information, and the mutation starts to occur. When the foreign parasitic DNA material interferes with the human own DNA, the proliferation and cellular division is potentiated with the aim of saving the own DNA and is discarding the invader's DNA. A spontaneous cancer remission is actually a longer process than considered initially, and is triggered when the parasitic infection has been completely healed. Dr. Weber demonstrated also very well that the disturbances of the mitosis in cancer cases are actually not endogenic and genetically controlled, but exogenic based on infectious degree of the hosting organism. The cells may divide, forming new multiple nuclei cells like in leprosy or syphilis. And when the body is depleted of nutrients, the mitosis abruptly stops and the cells are going to die. The brain, blood, and liver are the most resistant organs in this sense, and the kidneys and the lungs are the most exposed organs. And that's why you see much more cancer cases in those organs. First cancer manifestations after the CA protozoa start their four-stage evolution appears on the skin. So exanthema, for example, rashes, eczemas, and skin-dependent organs such as the mucosa membrane will show, or the epithelial inlayer, the hair and the nails, they all might show first signs of an infection. Acne might be a first sign of an infection with these parasites. Rosacea, psoriasis, scleroderma, syphilis skin rash, etc. At stage 2 and 3 of the parasitic infection, you might also have symptoms such as fever, night sweats, whereby the sweats are outside and inside of the body, affecting dramatically the different organs which are now becoming damaged by this infection. And Dr. Weber also found that the protozoa in all patients with polyarthritis, osteoporosis, rheumatism, kidney and liver disorders, lung conditions, dermatologic conditions, psychological as well as mental conditions was present. So these microorganisms, the microparasites, these protozoa, they can cause a whole range of conditions. Okay, so let's have a look at some video microscopy of the protozoa before we continue on with integrative cancer therapy concept that really works without side effects and that addresses and works exactly on all of these concepts that we've discussed. All recordings have one thing in common. You can see certain reoccurring types of microbes in all of them. There is no tumor tissue without these microbes. There is no blood of a cancer patient without those microparasites. The microparasites that have not yet been acknowledged by mainstream medicine as the cause of cancer have been comprehensively shown by Dr. Weber. They belong to two species. A. String shape, grown up. Their length is 2 times that of a red blood cell. B. Stick shaped. This is the most commonly found shape. The length of this gamete is about the diameter of a red blood cell. The youth form of both species are round or oval shaped. In normal blood tests, those parasites normally remain undetected, as they are hiding inside of the red blood cells. They are perfectly camouflaged by the exterior of the blood cell. The inner of the blood cell also offers plenty of food in the shape of oxygen and hemoglobin. The parasite enters as a so-called merocyte, the youth form, into the cell and fattens up and grows to its full size. Two gametes inside of the blood cell can be seen here. When the cell has been eaten empty, the sexually mature gamete leaves the cell and looks for a partner to exchange DNA with. Let's have a closer look at the parasites. We can see the stick-shaped A and the string-shaped microbes. Both species start off as tiny oocytes or merocytes. The youth form is called a trophocyte, which grows up to become a sexually mature gamete. A gamete can be seen here inside of a red blood cell. It excretes one merocyte after the other into the blood plasma. And here you see the string shape type B. Stick and string shape start off as so-called merocytes or oocytes. Here the trophocyte of the string shape type. This is the mature string-shaped parasite, a so-called gamete. When endangered, the gamete splits up in its survival form, merocytes. Here you can see this division of the microbe as a pearl-like chain. In this way, this gamete becomes again small merocytes, whereby each of those is the youth form of a gamete again. The legend for the following animation is as follows. Venous gas is depicted in blue, arterial gas in red, parasites are yellow, and the vacuole is a dark red. Here you see the mother cell with its cell membrane, the nucleus, and the cell plasma. The dots are depicted in the correct size relationship to the mother cell. The infection is first contained to the blood and lymphatic vessel system. Here we see a blood vessel with its walls and pores through which gases can be exchanged. It is surrounded by the connective tissue. Even though the blood vessels have pores, they only allow for fluids and dissolved matter to pass through. Even the youth form of the parasite is just too big to pass through. That means that a person is infected by those parasites but not ill yet. The defense cells of the immune system keep the number of parasites in check. Here you can see such a police cell of the immune system. You can clearly observe the so-called pseudopodia, the tentacles that are covered in sticky slime with which the leukocyte catches intruders. The coat microbes are then being transported into the innards of the cell and are thereby neutralized. It buzzes like a swarm of hornets in there, as you can see. If everything works as planned, this cell arrives at the liver with its prisoners, where it is chemically destroyed by huge eating cells, called macrophages, as can be seen in the animation. How big the burden of the infected blood cells become in the end can be seen here. Dr. Weber has added a chemical to the blood sample called b.a.r.t. The leukocyte tries to defend itself against this poison by creating an immense inner pressure so that it becomes a fully blown up balloon. We can clearly see the microbes caught in his innards. In the end, the cell membrane cannot cope anymore with the double strain of inner pressure and poison from the outside and bursts. And now you can see the dense cloud of microbes that swarm out. They sense a second chance and flee in all directions away from their prison. All recordings have one thing in common. You can see certain reoccurring types of microbes in all of them. There is no tumor tissue without these microbes. There is no blood of a cancer patient without those microparasites. The microparasites that have not yet been acknowledged by mainstream medicine as the cause of cancer have been comprehensively shown by Dr. Weber. They belong to two species. A. String shape grown up. Their length is 2 times that of a red blood cell. B. Stick shaped. This is the most commonly found shape. The length of this gamete is about the diameter of a red blood cell. The youth form of both species are round or oval shaped. In normal blood tests, those parasites normally remain undetected, as they are hiding inside of the red blood cells. They are perfectly camouflaged by the exterior of the blood cell. The inner of the blood cell also offers plenty of food in the shape of oxygen and hemoglobin. The parasite enters as a so-called merocyte, the youth form, into the cell and fattens up and grows to its full size. Two gametes inside of the blood cell can be seen here. When the cell has been eaten empty, the sexually mature gamete leaves the cell and looks for a partner to exchange DNA with. Let's have a closer look at the parasites. We can see the stick-shaped A and the string-shaped microbes. Both species start off as tiny oocytes or merocytes. The youth form is called a tropocyte, which grows up to become a sexually mature gamete. A gamete can be seen here inside of a red blood cell. It excretes one merocyte after the other into the blood plasma. And here you see the string shape type B. Stick and string shape start off as so-called merocytes or oocytes. Here the trophocyte of the string shape type. This is the mature string-shaped parasite, a so-called gamete. When endangered, the gamete splits up in its survival form, merocytes. Here you can see this division of the microbe as a pearl-like chain. In this way, this gamete becomes again small merocytes, whereby each of those is the youth form of a gamete again. The legend for the following animation is as follows. Venous gas is depicted in blue, arterial gas in red, parasites are yellow, and the vacuole is a dark red. Here you see the mother cell with its cell membrane, the nucleus, and the cell plasma. The dots are depicted in the correct size relationship to the mother cell. The infection is first contained to the blood and lymphatic vessel system. Here we see a blood vessel with its walls and pores through which gases can be exchanged. It is surrounded by the connective tissue. Even though the blood vessels have pores, they only allow for fluids and dissolved matter to pass through. Even the youth form of the parasite is just too big to pass through. That means that a person is infected by those parasites but not ill yet. The defense cells of the immune system keep the number of parasites in check. Here you can see such a police cell of the immune system. You can clearly observe the so-called pseudopodia, the tentacles that are covered in sticky slime with which the leukocyte catches intruders. The caught microbes are then being transported into the innards of the cell and are thereby neutralized. It buzzes like a swarm of hornets in there, as you can see. If everything works as planned, this cell arrives at the liver with its prisoners, where it is chemically destroyed by huge eating cells, called macrophages, as can be seen in the animation. How big the burden of the infected blood cells become in the end can be seen here. Dr. Weber has added a chemical to the blood sample called b.a.r.t. The leukocyte tries to defend itself against this poison by creating an immense inner pressure so that it becomes a fully blown up balloon. We can clearly see the microbes caught in his innards. In the end, the cell membrane cannot cope anymore with the double strain of inner pressure and poison from the outside and bursts. And now you can see the dense cloud of microbes that swarm out. They sense a second chance and flee in all directions away from their prison. New gametes are formed. Cancer according to Dr. Weber. Dr. Weber further inquired what would happen if the permeability of the blood vessels would change due to poisoning, radiation, or other stress factors. Wouldn't the result be that... Okay, so now that we have established that the cancer is most likely caused according to all of the research that we have on microparasitical infection, what is a cancer therapy approach that really works? Well, the cancer cells are always non-polar, as we've learned, opposite to healthy cells, which are dipoles. You can imagine then as a battery. Correct maintenance of the cell polarity is therefore required for the development and homeostasis of all metazones. Cell polarity mechanisms are responsible for the regulation of the asymmetric cell division of the stem cells, and disruption of the cell's polarity is a hallmark of cancer. Furthermore, recent evidence indicates that the loss of the cell's polarity is intimately involved in cancer as well. Secondly, cancer cells thrive through fermentation of glucose, as we've discussed already, and the metabolism of this primitive unicellular protozoa when they adapted from aquatic life, which is the lack of phosphorylation pathway, makes cancer cells crave glucose, and the lack of their energy is compensated by stealing up to 800 times the energy from the healthy cells. Thirdly, the genetic mutations are due to a DNA exchange between the protozoa and the human stem cells, as we've learned. And during the cancer tumor proliferation, you are not born with these genetic mutations and a predisposition for cancer, therefore. There is always also a protozoa reinfection risk, of course. The microparasites do not create immunity. So, what is our cancer therapy approach then? First and most importantly is that the cancer therapy must address the repolarization of the sick cells. This also implies an increase of the cellular membrane potential to 70 millivolts or higher. And this requires a lot of electrons being made available to those cells. Secondly, we must stop the fermentation by sugar starving and re-establish 36 ATP metabolism based on glycolysis and oxidative phosphorylation. This we do in order to correct and improve the mitochondria's respiration. Third, we stop the development of the cancer from a microparasitic intravascular infection in the blood vessels to a protozoa extravascular migration from the blood vessels into a connective tissue, and finally to a protozoa extravascular decompensated infection by invasion into the stem cells, and the cancer development as a protozoan infection cycle is very similar to malaria and toxoplasmosis, so we can use similar approaches here. When the microparasitic infection has been eradicated, the genetic mutations can be reversed through a healthy lifestyle and regular so-called matrix regeneration therapy. We can also, and we have to, of course, adopt a healthy lifestyle in general and regularly perform detoxification and disinfection procedures to prevent a reinfection and the further development of those microparasites. We should also avoid biopsies and surgeries if possible, because with those we increase the chances of spreading the protozoa infection faster throughout the body. And chemotherapy may work, but unfortunately the cost of chemotherapy is always decreasing general health, because not only those microparasites are eradicated and attacked, but also your healthy cells suffer. So a successful cancer therapy is very complex and must be a holistic and integrative approach that includes the whole body. It cannot be only targeted on tumor destruction. You must support the infected cells to get rid of the microparasites without destroying the healthy cells. The best recommended natural antiparasitic remedy is, in our opinion, vivid artemisinin, which is also the medication of first choice for malaria therapy, which is also, as we've discussed, a plasmodial infection, which is very similar to the cancer protozoan infection. With this kind of therapy, with vivid artemisinin, you do not destroy the infected cells to kill the parasites. You go selectively after the parasites, and therefore it's very efficient. As I've said, it's been recognized even by the World Health Organization as the most perfect treatment for malaria. And as you've seen, the plasmodial malaria infection is very similar to this protozoan infection that causes cancer. It really works. Vivid artemisinin is not just any artemisia extract like so many that you can find out there on the internet. It's much more concentrated. It's not oxidized, so we maintain the whole effects of the plant in this extract, and you will see it already from the color, which is a vivid green. That's why we call it vivid artemisinin and not brown like other artemisia extracts, because those are, so to speak, dead already. The ingredients have oxidized due to extraction methods, and this is not the case with vivid artemisinin. I'm going to post a link in the description box below. You can get it exclusively at lyra.me. Secondly, we need to detoxify and apply a milieu therapy to the body, especially for the liver, kidney, and the connective tissue. We must also purge out the microparasites, parasitical die-offs by regularly cleansing the colon, the liver, the kidney, the connective tissue, and here we recommend homozone, hydrocolonics, ozone insufflation, and ozonated oils. We must also boost our immune system using specific vitamins, minerals, enzymes, as well as a healthy nutrition, of course. And here we recommend that you supplement with magnesium, zinc, vitamin C, D3 and K2, as well as B12, B6, B3 and B1. We must also repair the cellular tissue and re-establish the correct functionality of the affected organs and optimize our pH and redox potential in order to increase the membrane potential. And here we recommend galvanic therapy with a great device such as the Veget Balance. You can also get the Veget Balance, of course, exclusively at lyra.com. Link to the Veget Balance also in the description box below. Then we must also energize the body, and we must boost mitochondrial activity to gain energy. We recommend that you do at least intermittent fasting, you exercise well, you use supplements such as L-carnitine, coenzyme Q10, L-arginine, quercetin, OPC, nattokinase, which is an antioxidant and blood thinner. And we also recommend, again, galvanic therapy for that with the veg balance. It's also great to meditate, to relax, to relieve the stress, to clean the active psycho-emotional conflicts and the negative state of mind out of our system. So forgive and forget, we call this, very important in order to stay balanced and to help your body fight off these microparasites. We should also avoid or diminish the electrosmog burdens. Here we recommend two great devices that are very inexpensive and help you with that, the Triamed CGI and the VitaChip, both of course available exclusively at lyra.com. We should also and must adjust the body's biochemistry and repolarize the sick cells, and this can be best achieved with the Geno 62 and again the Veggie Balance device. We also have to assess regularly the quality of the medication and the therapies received and correct the therapy approaches if the results do not show any improvement of the symptoms, and balance autonomic nervous system activity and a better quality of the bodily fluids and of the organs. And here we can use great devices such as the Kinlink Easy and the ANS analysis for studying the ANS activity to monitor live if and whether our medication and therapy approach is suitable for our patient and the conditions, and whether the patient responds really well to our approach or not. We can also check the state of the 17 most important organs with the ROFE system, and of course then we can adjust the nutrition to avoid allergies and intolerances, and here we can use EDS devices such as the Vistron, Easy Quick Chat and the new b.a.r.t system to achieve that. So as you see, the whole approach to dealing with this cancer problem is integrative. We cannot only go after the killing those microparasites, because those microparasites are everywhere and our body is actually capable of dealing with them in a certain amount pretty well if it is in a healthy state. And if you have further questions regarding all of this information, please do not hesitate to contact us. Shop at lyra.com. I'm gonna also post all of the complete contact information in the description box below for your convenience. We love to talk to you and advise you on what is best for you, your special condition, and the state of health you are in. And with all that being said, I hope that you've enjoyed this information. If you did so, please support our work by liking, sharing and subscribing. And until next time, stay healthy.